students, in this video we learn about blood physiology. So first let's see what are all the essay questions that was asked in your previous year university exams. How are leukocytes classified? Describe the morphology, properties and functions of each type. Define anemia. Classify the types of anemia on morphological and etiological basis. Define erythropoiesis. Discuss the stages of erythropoiesis. Discuss in detail the factors affecting erythropoiesis. What is hemostasis? Describe the mechanism of intrinsic coagulation. Add a note on bleeding disorders. Again, define hemostasis. Discuss the mechanism of clotting. Add a note on hemophilia. Describe the natural mechanisms responsible for arrest of bleeding. Mention the principle of two tests of hemostasis. Explain the intrinsic and extrinsic mechanism of blood coagulation and mention any two anticoagulants and their mechanism of action. Describe the mechanism of action of any of them. So let's see what are the short notes. Functions of plasma proteins, composition and functions of blood, ESR, anticoagulants, RH incompatibility, classification of anemia, erythroblastosis fetalis, ABO blood grouping system, megaloblastic anemia and pernicious anemia, lore of lymphocytes in immune functions, Landsteiner's law. So with a small introduction to hematology, blood volume. So 8% of the total body weight is contributed by total blood volume, and of this blood is made up of 55% plasma. So maximum contribution is by plasma, 41% by red blood cells, and nearly 4% by white blood cells, and 0.1% by the platelets. So in this chapter we are going to learn in detail about all of these. Now let's learn about hematopoiesis. What is hematopoiesis? The formation of blood cells from bone marrow is called as hematopoiesis, and this hematopoiesis takes place in the bone marrow in adults. What about the fetus? In fetus, from 3 weeks up to 3 months of gestation, it takes place in the yolk sac. From 3 months to the 5th month, it takes place in liver and spleen. From 6th month onwards, it takes place in the bone marrow. So look at these stages of hematopoiesis. You can see a hematopoietic stem cell which gives rise to all types of blood cells. Hematopoietic stem cells, they give rise to lymphoid progenitor cells and myeloid progenitor cells. The lymphoid progenitor cell, with the help of colony forming unit lymphocyte, it forms T lymphocyte and B lymphocyte. The myeloid progenitor cell, with the help of colony forming unit erythrocyte, it forms RBCs or erythrocytes. And with the help of colony forming unit megakaryocytes, it forms the platelets. And with the help of colony forming unit GM, granulocyte, monocyte, it forms the granulocytes, eosinophil, basophil, neutrophil and the monocyte. So let's learn in detail about erythropoiesis, which is a repeatedly asked long note. So erythropoiesis, formation of red blood cells is called erythropoiesis. And what are the stages in erythropoiesis? The first stage is the proerythroblast. You can see that the nucleus is very large here. The next stage is basophil erythroblast, which is otherwise called as the early stage. And you can see that this basophil erythroblast, it's called so because it stains with basic dyes. The next stage, polychromatophil erythroblast, it is called as the intermediate stage, and you find that the nucleus size has decreased and there is formation of hemoglobin. So hemoglobin begins to appear in this stage. The next stage, orthochromatic erythroblast. See, ortho means something correct. So orthochromatic means this has the correct amount of hemoglobin. So the hemoglobin is well formed in orthochromatic erythroblast. So this is called as the late stage. The next stage is reticulocyte. You can see that the cells have become smaller and there is no nucleus. Nucleus has disappeared. No nucleus. And these reticulocyte, they constitute of nearly less than 1% of the total RBC population. These reticulocytes are released from the bone marrow into the capillaries as mature RBCs, and these RBCs are biconcave disc-shaped structures. So this is how RBCs are formed, and the process is called erythropoiesis. What are the factors which affect erythropoiesis? Most important factor is erythropoietin. Erythropoietin is produced from the kidneys. So 90% of erythropoietin comes from the kidney and little is also produced by the liver. And this erythropoietin acts on the hematopoietic stem cells of the bone marrow to increase the production of RBCs and also hemoglobin concentration. And these RBCs travel with the help of hemoglobin throughout the system and they reach all the tissues and they oxygenate the tissues. So this is how erythropoietin increases erythropoiesis. And what are the other factors which increase erythropoiesis? See, erythropoietin is a major factor which increases erythropoiesis. The other factors are RBC and hemoglobin. So whenever there is a fall in hemoglobin or there is a fall in RBC, which is called anemia, so it will have a negative feedback and this increases erythropoietin secretion. So therefore anemia is another factor which affects erythropoiesis. Tissue oxygenation. So ultimately all this is happening to oxygenate the tissues. So when tissue oxygenation decreases, which is called as hypoxia, and when there is hypoxia, so automatically as a negative feedback, erythropoietin increases to increase erythropoiesis. So hypoxia is another factor which increases erythropoiesis. And hypoxia can physiologically also occur as you go to high altitudes or in any case of cardiac failure or lung failure. All this can result in hypoxia. So all these are the factors which affect erythropoiesis. Next, let's move on to anemia. Defect in RBC production is called as anemia. So this is a normal cell. Look at the number of RBCs. This is anemia where the RBC count has decreased. So anemia can be defined as decrease in the number of RBCs or even a decrease in the hemoglobin concentration. Both can result in anemia. How will you classify anemia? Morphological classification is based on the morphology, how it looks. There are three types where the RBC can look absolutely normal but still there is anemia. So this is called normochromic. Normochromic means hemoglobin is fine. Normocytic. Normocytic means the size of the cell is also fine. So an absolutely normal RBC but still the patient has anemia. So this can occur in cases of hemorrhage or any blood loss. The second type, macrocytic. Macrocyte means, as the name itself tells you, the cells are bigger in size. So bigger cells. And macrocytic anemia can occur in cases of vitamin B12 deficiency or folic acid deficiency, because vitamin B12 and folic acid are important maturation factors for RBC. If the RBC is not maturing properly, it will stay in a bigger size itself, resulting in macrocytic anemia, or even intrinsic factor deficiency. Intrinsic factor is produced by the parietal cells of the stomach. If intrinsic factor is deficient, B12 cannot be absorbed. So again, it results in megaloblastic or pernicious anemia. The third type, microcytic, as the name itself tells you, the cells are smaller in size. Microcytic. So usually it is microcytic hypochromic. Hypochromic means hemoglobin is less. as can occur in iron deficiency anemia or thalassemia. So this is how morphologically with the structure of RBC you will classify anemia. So the next type is etiological classification. So what do you mean by etiology? Etiology means the cause. So based on what is the cause of the anemia, again we are going to classify it etiologically. So in etiological classification of anemia, the first cause, bone marrow. Bone marrow itself is not producing RBCs. The defect is in the bone marrow. So when can you have a defect in bone marrow? Any patients on chemotherapy, radiotherapy, this could damage the bone marrow, thereby resulting in bone marrow damage and anemia. So this is called bone marrow deficient or aplastic anemia. The bone marrow is not producing cells properly. So second cause could be bone marrow is producing properly the cells, but the cells are not able to mature. Therefore, immature cells called macrocytes are accumulating. That's why it is called as macrocytic anemia or megaloblastic anemia, as it can occur with vitamin B12 deficiency or folic acid deficiency or intrinsic factor deficiency. The other cause is bone marrow is producing proper cells, maturation is also proper, but at the periphery cells are getting hemolyzed, they are getting broken down. This is called hemolysis. And all factors which cause hemolysis, they are called as hemolytic anemia, as it can occur with hereditary spherocytosis, any change in the shape of the RBCs, or sickle cell anemia, or erythroblastosis fetalis. All this can cause hemolysis, breaking down of RBCs, thereby resulting in anemia. And last cause, any chronic blood loss. Chronic blood loss. This can also result in anemia. Any chronic bleeding, internal bleeding, which we are not aware of. This also can result in anemia. So these are etiological classifications of anemia. Next, let's move on to erythrocyte sedimentation rate.